Okay, so now we are on to our third type of organic molecule, the nucleic acids. Many times when we do this with the first year class, we talk about nucleic acids and what they're composed of and a very general function of those. And then we take the time to go into the details of their structure and functions when we talk about hereditary material or the secret of life as it's written in chemicals. And so what we are going to do this time is go into a little bit more of those details and kind of combine both. So nucleic acids. Okay, so here's kind of the backstory. So we know that nucleic acids in general, they store and transmit hereditary information. Okay, and so we have three types of nucleic acid that we're going to look at. DNA, RNA, and actually ATP is a type of nucleic acid. And we typically do not talk about that in our first year class because we don't want to confuse students and have them associate ATP with DNA. There's 3 letters, it stands for something, and we just want to say ATP is cellular energy and keep it separate from DNA. But usually we'll have students who will notice that the composition is very close to RNA, and there's a reason for that. And obviously, they're nucleic acids. So here we go. So nucleic acids, we already just said in general what they are. The monomers are nucleotides. Each of these monomers has 3 parts to it. And if we make it bigger, and I'll have a larger version of that in just a second, it is a 5-carbon sugar. Oops, sorry. I'm trying to make that a little bit smaller. So a 5-carbon sugar. It's a pentose sugar because there are 5 carbons in that. We have a nitrogen base. And a phosphate group. So we call it the sugar, phosphate, and nitrogen base. And so a nucleotide has all 3 of those parts. Okay, so let's go over the general functions of these molecules, and then we're going to go further into the structure. So as far as composition of all 3 of these, we have CHOMP. Okay, so we have our carbon, hydrogen, and oxygen, which are parts of every sugar. The nitrogen base also has carbon and hydrogen and oxygen, but it has nitrogen in it. And then we have the phosphate that has phosphate and oxygen. So those are our 5 elements that make up nucleic acids. Nucleic acids, we know, are found in every single living cell at some point. And so we have DNA, which is the inherited genetic blueprints or instructions for building protein. And we think of protein, those are your traits. Okay, we know it's passed on, so it's inherited from either one parent if you're a product of asexual reproduction, or of 2 if you're a product of sexual. It's divided into genes, and each gene is a recipe for a trait. Now, we have some DNA we'll find that doesn't code for anything. It's kind of junk DNA, it's filler. But those that are coding sequences will translate into some type of protein. Okay, so DNA and RNA, we're going to actually do on a separate sheet. We're going to compare the 2. But as far as the function of RNA, it is there are three types. We have mRNA, messenger RNA, we have rRNA, ribosomal RNA, which makes up the bulk of ribosomes, and we have tRNA. Okay, all three are present during protein synthesis. So rRNA is part of the ribosome. tRNA are actually the transfer RNAs that build the protein while it's reading mRNA's instructions. Remember, in eukaryotic cells, DNA is locked up in a nucleus. So because of that, it doesn't direct the actual production of protein. It only provides the recipe for making them, for assembling the amino acids. So RNA does all the work. So RNA is ribonucleic acid. And it is active during protein synthesis. Okay, and we know DNA is deoxyribonucleic acid. And they get their names from the type of sugars that compose them. Okay, and we're going to go on a different slide and we'll compare the two. So let's go down to the bottom where we have an ATP molecule. So when you look at the ATP molecule, it also is composed of the same parts. So we have three phosphates, phosphate groups. We have a nitrogen base, which is called adenine. And we have ribose is the sugar. ATP is actually used for cellular chemical energy. And is produced even in photosynthesis. It's produced a little bit, but the bulk of it is produced from glucose during respiration. Okay, and that would be either anaerobic or aerobic respiration. It's used to drive mechanical, transport, and chemical work. So think about every energy conversion, everything that your body has to do. Its metabolism is how your body operates. It's its work. And what's interesting about ATP, we'll come back to it, is that the energy is actually found between the bonds of those phosphates. Okay. So let's go to the next one. Okay, so we mentioned CHOMP are the elements that it is composed of. Okay, so let's look at the differences between DNA and RNA. This is the whole molecular kind of looks kind of complex and kind of busy. But what you're going to notice about both of these is there's always a 5' end and a 3' end. Okay, and so when DNA strands are being synthesized during DNA replication, or RNA strands are being synthesized during transcription, so RNA is made during transcription. So this is the process that makes it. And this one is replication. Whether you're making RNA or DNA, it always grows in a 5' to 3' direction. So new nucleotides are always added to the 3' end, and you'll notice there's a hydroxyl there, and so again that will bond to a phosphate and we will get a bond, and that's how these nucleotides grow. So here's a nucleotide, here's a nucleotide. Okay, they both vary in sugar, and in these bases that compose them, and the number of strands they have. So we're going to look at that on a different slide. Oops, there we go. All right, sometimes you see the simplistic version of a nucleotide, and usually in the first year class we call the phosphate the sun, we call the sugar the house, and then the nitrogen base is the garage. Okay, so all nucleotides have those three parts. So let's look a little bit further into what those bases are. So the bases that make up, this is the language that DNA is written in. Okay, so this is actually the variable part of DNA. Okay, this is the language of genetic material. So what makes you different in your inherited genes is the arrangement of these bases, not the sugar phosphate backbone, because that is consistent with every nucleotide. So everyone has the same sugar phosphate backbone. It's just the arrangement of these bases, the order of those bases is what gives you your uniqueness, but also your sameness. So if you share different traits with each other. So for DNA, there are four of those bases. You have A, G, C, and T. And the reason they're listed this way, usually we say, well, A and T always bond together, North Carolina A and T. There is a Amoeba Sisters episode that says apple in a tree, car in a garage. And I usually say North Carolina A and T, U and C, G. Okay? But you'll notice that the A, adenine, and guanine both have 2 rings in their sugar. Okay, so 2 rings of sugar, and that makes them purine bases. C and T only have 1 ring. Okay, and because they only have 1 ring, then they are pyrimidines. So 1 ring of carbon and nitrogen here, and we have 2 rings with the purines. So a purine always pairs with its mate, which is going to be a pyrimidine. Now again, this is in DNA because DNA is double-stranded. And you can see this would be that DNA. So we know that DNA is double-stranded. It has 2 strands of nucleotides. And they are called anti-parallel. And the reason for that is it looks like one side is upside down. So one side runs 5 to 3, okay? The other side runs 3 to 5. But you always add to the 3' end, okay? So they're called anti-parallel. One runs one way, one runs the other way. And this shows the bonding. What happens here in between are hydrogen bonds. So again, the same hydrogen bonds that hold water molecules together, the same is true for these bases across from each other. So you have your A and T, C and G. There are 2 hydrogen bonds between A and T. There are 3 hydrogen bonds between C and G. The backbone is called a sugar phosphate backbone. Okay, so if this is a straight ladder, the rungs or the steps in the ladder would be your bases. All right, so specifically talking about bonds. So if carbs have glycosidic bonds, covalent bonds, lipids have ester bonds. These are called phosphodiester. And so even though they're made, these polymers, again, are synthesized with dehydration synthesis or a condensation reaction. So that means water is released when a nucleotide is added to another nucleotide. So when we're copying DNA or RNA is being built using DNA's instructions, that's when this happens. So you can tell that the phosphate, the OH part, the hydroxyl of the phosphate, is going to attach to a hydroxyl of the sugar, okay? And when that happens, the bond that links them together here is called this phosphodiester bond, and water is released. So let's kind of recap. You've already heard some differences, you've heard some similarities. Instead of going through this like that way, I want to do kind of a Venn diagram. So let's do DNA versus RNA. Okay, so we know they're both nucleic acids. They're both composed of nucleotides. That means they both have phosphate that's exactly the same. They both have a sugar, and they have some nitrogen bases. The thing is, the sugar's different. Okay, so we have ribose on the RNA side, and we have deoxyribose on the DNA side. Phosphate's the same. They're both nucleic acids. They both have strands. But let's go down to, before I do that, let's do the nitrogen bases. So the nitrogen bases that are the same are A, adenine, cytosine, and guanine are the same. The difference would be where DNA has a thymine, you might remember that RNA has a U, a uracil. Okay, and DNA is 2 strands of nucleotides, and this is 1 strand of nucleotides. And with DNA, we know that it's in a double helix. So it's a twisted ladder or a spiral staircase. Okay. One more thing I want to just go ahead and throw out while we're at it. RNA, we need to say, is going to help assemble proteins. It carries its instructions, so protein synthesis is its thing, and DNA is just going to hold the instructions for that. So they're both involved in protein synthesis, but RNA is more directly involved, and DNA just holds the recipes. All right, and the last nucleic acid is adenosine triphosphate, ATP. And so you can see here is what it stands for. It is considered a nucleotide. We said it has 3 phosphates here. And the energy is found in these bonds, okay? So normally in the first year class, we'll go A, P, curvy line, and those curvy lines represent those phosphodiester bonds. And those bonds, that's where the energy is stored. So when they're broken, energy is released. And this phosphate then goes and attaches to whatever needs that energy. So it's kind of the energy currency of cells. And that's the nucleic acids.